Hello, everybody. We're going to get started in about a minute. We're just going to let everybody come into the room.
Hello.
Okay, I think I'm going to get it started. So, good afternoon, everybody. I'm Maya Kadar Kovalsky. I'm a governor at Tel Aviv University and a board member of American Friends of Tel Aviv University, where I chair the Alumni Committee. And I am also a very proud alum of the university. I am delighted to welcome you to today's webinar, which is a one-on-one conversation between these two women right here, who are Tel Aviv University trailblazers. Both of our guests are accomplished experts in their respective fields. They are leaders, inspiring, interesting, and bold. I am speaking about none other than Dr. Tali Ilovitsh, who is an assistant professor in the Department of Biomedical Engineering, and Sagi-Maydan, who is a journalist and Tel Aviv University alum, and also one of my fellow board members on the American Friends of Tel Aviv University board. So during the next hour, you will have the opportunity to get to know Dr. Ilovitsh, the researcher that Yitzhak Herzog, the president of Israel, visited when he was on campus last month. Dr. Ilovitsh leads a translational research program that focuses on using ultrasound and nanobubbles to destroy cancer. She pioneered optically inspired ultrasonic techniques to establish new imaging methods, and her innovative approach is a medical breakthrough, which is not surprising for a woman who has been named one of Israel's 30 under 30 by Forbes Israel. And now I'm going to turn for a second to Mary Sagi-Maydan, who is a journalist, architect, and Tel Aviv University alum. She is an accomplished journalist and was the former U.S. correspondent for the prestigious newspaper Globes and Haaretz in Israel. She has interviewed some of the most inspiring figures of our time, including Mikhail Gorbachev, Barbara Walters, Larry King, and Jerry Seinfeld, among others. A passion for design and a clear vision of what she wanted her home to look like led her to her next career. Mary is the founder of Maydan Architects, specializing in modern design. We will begin with a conversation between Mary and Tali, followed by a Q&A from anyone who is watching. If you have questions, please type them in the Q&A box, and we will do our best to get to as many as possible after their one-on-one conversation. So now it is my pleasure to turn the program over to Mary and Tali. Enjoy, guys.
Hi, everyone. Thank you, Maya, for the nice introduction. You know that I want to do a webinar with you one time because you're not just a successful businesswoman, but your impact investing and your philanthropy is inspiring. So you're next. And Tali, it is such a privilege to sit for a one-on-one conversation with you. I am inspired by you.
Thank you.
Meeting you in Miami was amazing. You have an incredible research going on. And I want to start with a little anecdote. So I understand that I was told that when you completed your postdoc at Stanford, all the universities in Israel offered you a position, and you chose Tel Aviv. And Professor Karen Avraham from Tel Aviv University, from the med school, the dean of the med school, told me that at some point after signing with Tel Aviv, it seemed like everything might crumble because of mice. So it's a really nice story. Can you tell us about what happened there?
Yes. So first of all, I'm really happy to be here and have this webinar. And going back to your question, when I completed my postdoc and I was looking for a faculty position in Israel, the situation here, at least in terms of the faculty of engineering, are such that usually animal facilities are not in close proximity to the faculty itself. So animal facilities are usually either at the med school or some other areas, which are usually far away from engineering. And in our research, because we are trying to cure cancer or trying to treat different diseases, one of the things we have to do is we have to have experiments in an animal model. In our case, it's usually mice. So for us, it's crucial to be in close proximity to an animal facility. And in most of the faculties of engineering in Israel, the animal facilities are far away. So when I arrived to Israel, it turned out that here it's a similar situation, and I was unable to start having experiments. And Karen, what she did when she heard about the situation, she said that she will give us an area, a space in the animal facility and organized it, despite the fact that I'm not directly linked to the Faculty of Medicine. And she said it because she said that I'm a faculty at Tel Aviv University, and although not in her faculty, in her department, but she still did it. And this is only one example of really how Tel Aviv University faculty and other members support all of the faculty members here, which is really inspiring.
So the mice are comfortable. You have your own very large lab. You a few years ago named 30 under 30 by Forbes. What does it mean to you, the 30 under 30?
So first of all, I was really excited to hear about it. It's not something that, you expect. I think that my vision is really to try and develop technologies in the field of ultrasound in order to treat diseases. And having accomplished or having named 30 under 30 is just one thing along a line of my vision or what I truly want to accomplish, which is really getting to the clinic and trying to help people and treat them with the technologies that we develop. And I think throughout my career, I was always doing things ahead of time. So I started a master's degree that was in a direct track in parallel to my undergraduate degrees. And I kept trying to do things really quickly. So my postdoc was a little over 2 years. And that's why I was able to achieve everything at an age of below 30, unlike other students, I would say, here in Israel, that usually because they join the IDF, they start to study a little bit later, and also they have breaks between degrees. So I always try to do things very quickly, and fortunately, I was successful in that. So this is where we are.
I have to ask you about that, and then we'll go back to 30 under 30. But you said about doing things quickly in research and being a scientist. I always think that you need to be very patient because things are slow, isn't it?
So usually it is true, especially when it comes to trying to treat diseases and biological conditions, because when it comes to this, you have to have a model and you have to try it. And it really depends whether you have a way to test it, not only in mice, but also to try it in other methods. So one of the things that we do is we develop physical technologies. We didn't talk about it yet, but one of the things that we develop are these bubbles that we just use them as detonators to implode cancer cells. So in our case, what we can do is we have the physics, so we have physical modeling of these bubbles and how they respond to ultrasound. We then have experiments that we do in cell cultures, and we see that it works. So the leap into doing experiments in mice, in our case, is not as huge as starting only in mice and trying to identify something. So for us, because we are doing it really systematically and we test our methods before, it usually increases the ability to publish and disseminate the findings of the study.
We'll go into the research in more detail soon, but I wanted to first ask you about go back to your being so young. So how does it play out? And I know that sometimes people, even your students, mistake you for, are you the professor? You're so young.
It used to be, you know, as I grow older, it's not as much as it used to be. But I think in some ways it's an advantage because I think that I'm really curious and I'm really open to new ideas. And sometimes, you know, when you have younger generations, you need to be somewhat in their minds. So I'm still considering myself at an age where I'm able to do it, despite the fact that sometimes, you know, I'm still, I see the students and I think to myself, wow, did I act like that when I was that young? But it still happens. And sometimes I need to really prove myself in order to other people to understand that I'm not as young as, or that I don't behave as young as maybe I am. But like I said,
Give me an example. What do you mean? Prove that you're not, you don't behave as young.
So sometimes when I sit with other panelists in like a panel where we need to review proposals, people look at me and they're like, oh, what are you doing in the panel? So I need to introduce myself and I need to say that I'm a faculty member. And then when I start explaining the justification for example, the ranking that I give a proposal, then they understand, or at least I think they realize that I'm somewhat equal. So sometimes it happens. Usually it's not the case, but I think it happens to a lot of people also.
And you told me a nice story that you were congratulating a student of yours. That was, you went to the parents. You can tell it better than me. It was such a nice.
Right. So one of the things that I'm really proud of are my students. And my students, I keep encouraging them to apply to different scholarships. And a lot of my students get scholarships. Just today, a student of mine got a scholarship for the treatment of of Parkinson's disease by the APPD Institute here at Tel Aviv University. And so about a month ago, we had an event where one of my PhD students received a scholarship, and we went to the event. And I went to the family that donated this scholarship and I introduced myself, and they thought that I was the recipient of the award because I'm the student. And I explained to them that I'm the head of the lab, but still they still said, here's the PhD student that gets the prize. So it's still, it's a nice confusion though.
So you're not only a brilliant scientist, you are also a mom of four. And you know, Michelle Obama once said, I can do it all, but not at the same time. Well, I have to ask you for your secret.
I think there isn't a secret. I think that you just need to work really hard, and you need to find a somewhat balance between life and work. And what I at least do is I try to be there for my kids in terms of every day. I usually try to pick them from school and then be with them until they go to sleep. And then I continue to work after they go to sleep. So I try to maximize the time that I spend at work and also with my children. So I will be an engaged parent. But it is hard. And I think it gets a little bit easier as the kids grow up. But when they were younger and we moved to the US, to Sacramento with my three kids. At the time, I had three children, and all of them were under three.
All were under three, right?
Right, all of them were under three. That was really hard. But still, you managed to do it just by balancing. And also, I really have a lot of support from my family and my husband as well.
So you mentioned the sleep deprivation. Other than sleep deprivation, what are really the skills that you think are crucial to run such a successful lab?
I think that like everything, it improves as you practice, but to run a lab, you need to be really multidisciplinary in the sense that I need to recruit funding because our experiments are big and they are costly. We need to use a lot of consumables, and we use, like I said, mice, and we have all of the ultrasound equipment. So for all of that, I need to recruit funding to be able to do it. So on the one hand, some of my time goes towards that. On the other, the students really need guidance and mentorship. So what I do is I have like an open door policy that I'm here in my office, and when a student is having an experiment and something is going wrong or he wants to consult about something, he just comes, and if I'm available, I will answer immediately and try to guide them. So you need to be a good mentor as well. And also you need to prioritize as everyone, because sometimes I would have a clash of things and I'll have to choose what to do. And this is just how I do it. So, and also you need to pick the right team. That's really crucial because really the students at the end of the day are the ones who are doing the research. They're in the lab, they're doing the experiments. And fortunately, I had such amazing students. I think it has to do obviously with the fact that I'm at Tel Aviv University and we have the best students in Israel, possibly worldwide. And really, they are so mature in the sense that they come, they have ideas. It's not like I'm telling them, do something and they do it. They would ask why, they would suggest how to do it in another way. They come up with new ideas as well. So the team is really important. Also, I have an administrative assistant that she's amazing, and a lab manager that he was a PhD student in the lab, and once he graduated, he became our lab manager. So all of this team together is what helps us to really excel and have our research progress.
You mentioned a lot of different things that you have to do. What is your favorite? Where is your happy place?
My happy place is to be in the lab and to do the experiment by myself. This is something that I am always happy to do because I'm a person who really likes to work. And sometimes, when you need to raise funding, so you need to write grant proposals, etc. But usually what I try to do is to have a few hours every week. In the past, it was a full day, but now it's a few hours that I go into the lab. I prepare microbubbles. I do an experiment with the ultrasound. I set up some kind of another setup. And this is where I'm really happy.
What about the raising money? Is this a difficult part of the job?
I think raising money is really something that you also learn, and it's an art. But what makes it easier, at least in my view, is the fact that our technologies are truly aimed to save lives. And when I write it and I explain why and how and what is the impact, I think it helps because in terms of receiving grants, I received recently an ERC starting grant, which is the highest grant that a young faculty member can receive it's by the European Union. And after receiving that, I was like so happy because it was for treating a very devastating brain tumor, glioblastoma. In here it's in mice, but we are doing it with the technology that we developed with the nanobubbles and the ultrasound. And it also gives me some kind of a confirmation that other scientists and institutions believe in what we do. So I think overall, fundraising, it's not fundraising, it's grant proposals are something that are very competitive and challenging, but I do my best in writing them. And so I think our situation is pretty good in that sense.
So speaking about money, you did your postdoc at Stanford. When you look at those labs and institutions with these kind of endowments, how can Tel Aviv University compete? And we are competing really well, but how do you do it?
I think when I was a postdoc, we had an amazing lab, really. It was a multidisciplinary lab, and I think the lab was of Professor Catherine Ferrara at UC Davis, and then later at Stanford University School of Medicine. And there is where I really studied how to do research and how to do good research. And she had an amazing lab with so much things. For example, I'll just give an example. She had an MRI that was almost solely was used by her lab. So we had an ultrasound device that could enter this MRI and we could use it. And this is something, in Israel and even elsewhere in the US, a lab does not really have its own MRI. And when I thought about moving to Tel Aviv, because for me, I was sure that I'm not going to stay in the US, I really wanted to go back to Israel. And so when I wanted to come back to Israel, I knew that having an MRI is something that I will not get. So to begin with, I decided to go for research that will not involve MRI. So in terms of other things that we use, we have programmable ultrasound systems that are pretty expensive. They're about $250,000, but it's not as expensive as an MRI. And Tel Aviv University, when I arrived, they gave me like, how you call it, like funds to establish my lab. And so in terms of the funds that they gave me, they got me a really good package, and I was able to purchase everything I wanted to start my lab. After that, I need to raise the funding by myself. But at that time, I had everything I needed to start my lab, aside from an MRI, but we can't ask for all. So I think that in that sense, Tel Aviv is really competitive, even compared to other universities in the US, for example.
Is there a difference in the work culture between Israel and the US?
I think that in the US, it was more regulated, I would say, in time. So there were like times where the lab, most of the people arrived at 9, left at 5. In the US, it's more flexible. Some students will not come at 9 because it's too early for them, and they will arrive later and stay later. Also, in the US, sometimes you have really specific routines, even though in the lab that I was in, this is one of the reasons that I chose this lab, because as a mother, I had to do this balance between work and family. And when I was in the US, on Fridays, I always wanted to leave a little bit earlier. And so my mentor there, she was okay with that. The lab was also of working mothers. And this is something that is not as frequently seen in other labs, because in other labs, I've seen that people work also on weekends and things like that. And here in Israel, each lab is more independent. So in that case, I set the time for the students, and I'm really flexible, similarly to what I wanted to have when I was a student. So here it's more flexible, I would say.
So let's go a little bit back to the beginning, and I want to start as a kid in Israel. You're a gifted kid, and did you always want to be a scientist? What steered you towards it and towards your research?
So I always liked math ever since I was really small. And at the time, in this gifted children program, we had a class where we learned how to build rockets. And we even had to fire them, which was really nice. At the time, everything was fields. You didn't have so many buildings. So we could go there and just launch the rocket. And I was really, I really remember that I enjoyed doing it. And this is one of the things that led me afterwards to become an engineer because I like to build it and to see how it flies. And at the time, because I liked physics, I liked math, when I wanted to go to the university, I thought about something that combines both and decided to go with engineering. So I guess that I was attracted to it since I was about 10.
And when did the interest in cancer start?
Well, that started later, much later. So when I started, I studied actually electrical engineering in all of my degrees, because at the time I didn't want to study anything that is linked to medicine. And I think during my master's, I worked on something that was completely electrical engineer. It had to do with building these modulators that should have gone into computers to try and create a computer that works in the speed of light. And it was really engineering. But for my PhD, I transitioned a little bit to microscopy. And microscopy is still optics, similarly to my second degree, my master degree. But microscopy, you need to image something that is biological. In that case, it was cells. So we image cells, and I developed super resolution techniques to try and image the cells better, to have better resolution, to see finer details in the image. And so this was like my first touch with biomedical engineering, even though it was electrical engineering. And then towards the end of my PhD, while I was working in optical imaging and microscopy, I ran into a paper that implemented the same exact concept in ultrasound. And I was amazed by that because by the time I had kids and I knew ultrasound, I was familiar with it only throughout my pregnancies. And always I saw these images and they look really bad. You know, they're grayscale and dotty and you don't know exactly what you see. And I figured that if there's a way to improve ultrasound imaging and get better results, it could help save lives because ultrasound is the most frequently used medical imaging modality nowadays in the clinic. And it is safe. It does not involve ionizing radiation. It's cost effective. And so I decided that I would like to transition to that field, to the field of ultrasound. And this is how I moved into biomedical engineering. And actually, I had to find the lab without any experience in ultrasound imaging. Thinking about me now, if I would get an email from someone saying, hi, and I would look in his CV and I will see that he has no experience with ultrasound, that could be a bit risky. So I sent applications to multiple labs worldwide for my postdoc. And fortunately, I also emailed Professor Catherine Ferrara, and I told her that I have a lot of ideas that come from the field of optics that I would like to implement in the field of ultrasound. And she agreed, and that's why I moved with my family to her lab. And luckily, we were successful because the physics is the same physics, or it's similar physics. It's a wave physics. Microscopy and optics is a wave. Ultrasound is a wave. So the physics could be implemented in both. But when I came to my postdoc, my ideas were in the field of imaging, so trying to improve imaging. Kathy's lab was also doing a lot of ultrasound therapy. And this is where I got exposed to ultrasound therapy and fell in love with that field as well.
So it's a good time to dive a little bit more into the research in some more detail, but simplified so we can all understand.
So what we do in our lab is we usually use ultrasound contrast agents. So in almost every medical imaging modality, there are contrast agents that aim to enhance the images. In MRI, for example, we have gadolinium that you need to drink or they inject it before you do the exam. In ultrasound, the contrast agents are called microbubbles. So microbubbles are tiny, tiny bubbles that they're about 1 micron in diameter, 1.5 micron, which is 1/10 of a hair or 1/10 of a red blood cell. So similarly to red blood cells that can circulate freely in the body, in the bloodstream, microbubbles can do that as well. And they are covered with a material that prevents them to aggregate, so we don't get a big bubble. They remain tiny. They are cleared from the blood circulation about 10 minutes after we inject them, so they are fully biocompatible. And what they do initially when they were developed as contrast agents in order to improve the imaging of blood vessels. So you would be able to clearly see them. They are approved as contrast agents in ultrasound for, I think, over 30 years in the clinic, but they can also be used for therapeutic applications. Why? When we apply ultrasound to these bubbles, ultrasound is a pressure wave. What will happen when you change the pressure that you apply to a bubble? So if we'll think about a balloon that we start to blow air into, what we'll get is that the bubble will start to expand and contract. So we get these oscillations. With these oscillations, we can achieve bioeffects. So we can have therapeutic effect in the body. How? For example, we use very small bubbles. They're called nanobubbles that we inject them to the bloodstream via an infusion. And then they accumulate within tumors. And when they accumulate within the tumor, we apply the low-frequency ultrasound. The low-frequency ultrasound is focused to the tumor region. The bubbles, instead of expanding and contracting, we apply ultrasound in a manner that causes them really to fragment or even explode. So in that way, we can fractionate the tumor, and it is an alternative to an invasive surgery. So we can do this treatment instead of doing a surgery when we want to remove the cancerous tumor. So this is one example. Another a very promising application is for brain therapy. What's interesting about ultrasound is that at specific frequencies, ultrasound can actually penetrate through an intact skull. So the ultrasound waves penetrate through the intact skull, and we can focus them similarly to the way we focus light with magnifying glass. We can focus them deep inside the brain and have therapeutic bioeffects in the brain. One thing we do with our microbubbles is we are opening the blood-brain barrier that exists inside our brain and facilitate the delivery of different drugs that otherwise cannot enter the brain. So for a variety of brain diseases. And these are just two examples.
Does it work mainly on a mass that is not diffused, or can it work also on cancers that are diffused?
So what we do, or at least what really our hope is that we could also treat metastatic tumors. What happens is that similarly to surgery, usually you would resect the primary tumor. But if the tumor has metastases, then not always you will be able to remove each of them. What we've discovered recently is that when we apply this treatment inside the tumor, what we get is also an immunological response. The reason cancer grows is also because it can evade the immune system. So the immune system does not recognize it, and therefore it can grow. But with our treatment, because we are causing this mechanical damage inside the tumor, we see that the immune system is being recruited and it comes to the tumor to see what's going on. And also when we work in a model that we have multiple tumors and we only treat one tumor, we see an effect on a distant tumor. And this is really promising because in that way, if we could treat metastases, this would be a huge breakthrough. And this is our hope. And also similarly to surgical resection of tumors, usually it's not sufficient to do that. We would like to combine it with either chemotherapy, or in our case, we combine it with immunotherapy to further enhance the immune system to attack the metastases as well.
So you've mentioned glioblastoma, and I have been following the research on glioblastoma for many years because my beloved mother-in-law died of glioblastoma. Do you tell us where it is with glioblastoma? What is the status of the research? And do you think that it can be curative or just extend life? What's your goal? What do you see happening?
So specifically glioblastoma is a really aggressive tumor that there haven't been a lot of successes in the past 20 years or so, and the conventional treatment remains somewhat the same. There are some limitations. One of the limitations is, like I said, the blood-brain barrier that exists inside the brain. And this barrier prevents the delivery of different chemotherapies even to this tumor. So what they do nowadays is they inject systemically a chemotherapy that can penetrate a little bit or somewhat well this barrier, but it's not necessarily the ideal chemotherapy for this tumor. And they just do it because this is something that can penetrate the barrier. So with the technology now, where the BBB can be opened safely with bubbles and ultrasound, it is feasible to try different drugs that were developed in dishes, in cells, and could not have been tried in vivo otherwise. So there are a lot of promising candidates, but it needs to be still tested. And there are a lot of clinical trials that are going on. There aren't a lot of success yet. So currently the aim, because it's such an aggressive tumor, it's mainly to make this a chronic disease. So it's not to cure this GBM, but it's more, like you said, to extend life. But if you extend it in a significant manner, it's still a huge advancement compared to what currently exists.
So, you know, I was standing next to you in Miami at the conference of Tel Aviv University when, after your presentation, someone from the audience came over to you and said that his friend was diagnosed with glioblastoma. He has a few months to live. And is it possible to try your treatment because he has nothing to lose? I guess you get a lot of those requests. How do you feel about it?
So every time I get such a request, first of all, it's really heartbreaking because I know how hard it is to try and translate studies that are currently being done in mice to the clinic. So it's not like I can now take the system that I'm working on with mice and just do the translation to humans. So within a time frame of a few months, it's not really feasible. But my really hope is that we could establish an ultrasound department or an ultrasound institute at one of the hospitals in Israel, where we could translate our technologies in that institute and really start trying them in humans and having the impact on patients as well. So this is currently what I'm trying and hoping to achieve.
Do you feel sometimes that you're in a race against the clock?
I think always, because since our technologies are working in mice, at least some of them, and I know that if we could have done it in patients, it could help save lives, but we are doing whatever we can in terms of the time that it takes, the fundraising towards this ultrasound institute that we want to establish, and also in terms of regulation, because it's not like now, even if I have an amazing technology, I can't just do it on patients. I have to follow the regulation, I have to get the approvals. So it's always a race against time, but we're trying our best to make it happen soon.
What's your biggest dream?
My biggest dream really is to see our technologies being implemented in the clinic. That would be the vision. That's what we want to accomplish. And we want to see patients that are enjoying these technologies, because one of the things that I truly believe in is that in cancer in general, there are so many types of cancer. And when you develop a biological treatment for cancer, the biological treatment has to be tuned to the specific cancer type. And here with our technologies, they are physical technologies, meaning that the bubbles, when we cause them to explode and fractionate the tumor, if it's, for example, a breast cancer tumor, it doesn't matter if it's HER2 positive or HER2 negative, it will destroy this tumor. So I believe that these technologies, the physical technologies, are the ones that could have a big impact and could be implemented in a variety of different diseases as well. So that's my hope.
What's your biggest challenge?
The biggest challenge is what we discussed, is how to translate them into the clinic. And for that, we have to recruit and build better instruments, and instruments that are compatible to patients and not just the ones that we are using for mice. And so this is a major challenge. And if we could overcome this, we will be able to hopefully translate our studies into the clinic.
So, I'm sure that every patient or a relative of a patient listening to us wants to know, okay, so what does it mean in terms of time? When do you think we can, realistically, it can translate?
Each technology is a bit different. So, for example, nowadays, in terms of brain therapies, there are some clinical trials already about blood-brain barrier opening with microbubbles. It's not the same nanobubbles that we are using in our lab, but there are some really nice results with current clinical trials. But in terms of what we do, for example, for breast cancer, we are now in the process of obtaining the approvals to try and go into phase 1 clinical trial. And it could take, let's say, time frame of, say, 5 years. I hope to see it already in the clinic. There are other studies that we are doing that might take a little bit longer. But in terms of time frame, even 5 years is fast, given the fact that we only started these studies a few years ago.
Your work in the field is so amazing. I want to ask you, it seems when I read your biography that you went from success to success to success. Is there anything that you consider unsuccess?
I think that you learn from everything. And so even you can take something that was not as successful and make it a success looking back. So like I said, during the course of time, when I started my master's degree, I didn't really know what I want to do or what I like to do as a researcher. Like I said, at the time, I didn't even think I want to work in the field of biomedical engineering. I think at the end, if I knew that this is something that I like, I would have done it already for my master's degree. So this is one example for you.
You still remain so modest. You sit with a lot of egos in meetings all the time. What's your strategy to handle it?
So when I was doing my undergrad degree in electrical engineering, we were about 150, and only 6 were girls. So I guess I'm always used to being with a lot of men in the room. And usually I would say that still it is the case even now. When I sit a lot in committees and even in terms of faculty, there are still differences. So I guess that from very young age, I'm used to being like the only woman in the room, and you just learn how to deal with that, and it's okay. I'm used to it. So I don't have like a trick or something. It's just something that you learn how to do.
Who is your role model?
I have a few. The first model was my mother. She's still a computer programmer, and she studied computer science in an era where there was almost no computers. And she always liked sciences, and she really encouraged us to see that, to proceed with what we like in that field. And I'm also, another role model is Marie Curie, that I also think of her when she was working in an era that was dominated by men, and she was really a trailblazer. So I think these two.
So we have very little time left for a couple more questions, and then we'll hand it over to Q&A. But what's the biggest misconception about you, do you think?
Oh, that's a hard question. I don't know. I think that it's really what you see is what you get. So I don't know if there is a misconception about me, or at least not that I'm aware of.
And I like to always end with a question that Baba Walters asks. So complete the sentence for me. Tali Ilovitsh is. That's a hard one, I know.
Yes, that's a really hard one, but I would say that Tali Ilovitsh is always curious, because ever since I was little, I'm really excited about things and I like to study them.
So you're always curious. I think that Tali, if I had to say, I'd say Tali Ilovitsh is a rock star. And now we'll hand it over to Maya for questions from our audience.
I concur, a rock star indeed. I wanted to, I'm hopping on because we only have a little bit of time left, and I wanted to make sure that we answer. We got one question in the Q&A, but also I have a few of my own questions that I might also add. But anyhow, I wanted to know for the person who wrote in, can you speak to the research and collaborations and vision to bring it to market? So I think you might have covered this a little bit, but why don't you go over it again?
Our research is really a research, or our technologies are platform technologies. So for example, when we open the blood-brain barrier, we have this technology, and now we can deliver different drugs. So for example, we have a collaboration with Teva Pharmaceutical Industries, and also with the lab of Professor Dani Pe'er, who is the Vice President of Research and Development at Tel Aviv University. So Dani's lab is developing lipid nanoparticles for the treatment of glioblastoma, as an example, and we have the technology to deliver it across the barrier. So we are collaborating through the support of Teva and the Israeli Innovation Authority in order to deliver these lipid nanoparticles to treat glioblastoma. So we have a lot of collaborations. Also, yes, I see a question about Insightec. Insightec is a leading company here in Israel. They are developing MR-guided focused ultrasound systems, and we have a collaboration through a sponsored research with them. We had it for 2 years. So they are a company really that has FDA approval, and they do the treatment in patients in Israel and worldwide in terms of brain and other organs as well. So they have these systems, and we have a collaboration, and like I said, a sponsored research with them about studying delivery to the brain.
Does your lab also have any relationships with hospitals?
So what I do is I have collaboration with clinicians in order, for example, to guide us in terms of if we look at immunological effects that we are trying to trigger, if we want to combine our treatment with immunotherapy. So we always consult with clinicians to be clinically relevant and understand the field, because like I said, in my background, I'm an electrical engineer. So I always like to collaborate in order to fill the gaps and have our and be clinically relevant in terms of our studies.
Okay, we have Gary writing in that most importantly, how do you balance raising a family, husband, and being Orthodox with your work? Yasher Koach for all you do. So I also agree for that. I feel like you spent some time speaking about that. But if you want to say anything else, you may add.
I think that it's just like I said, you have to balance and you end up not being very satisfied with either because you always feel that you're missing time, that I don't spend enough time with my family or I don't spend enough time at work. But like I say, I try always to do the best I can, so I can do better than what I try.
Exactly. I think
Do you
that me and Mary can sort of understand you there, right? We feel you. Do you want to
Are your
add
kids
something?
fascinated by the research? What do they think? Are they interested in it?
Yes, so my kids don't understand how I'm Dr. Tali Ilovitch and I'm not a clinician. So to them, it's like a mystery. And when I tell them also that we are developing these technologies to try and treat cancer or the brain, they're still puzzled by how come I'm not a doctor. But they're very interested. My kids are also enrolled into the gifted children programs. And so I really hope that we are raising the next generation of future scientists in our house. So hopefully.
Here, here. I wanted to ask a question. This is now my question. So how much does an MRI machine cost?
I'm not sure, but I think it's a few $1,000,000.
Okay, got it. I wanted to just understand if it was within the realm of, possibility at all.
So it's something that, there are some donations of MRIs at hospitals in Israel, but it's an expensive donation.
Okay. I wanted to understand, there's one thing that, you spoke about ultrasound and how you're working to make it better. But can you take us back a little bit about and tell us a little bit about ultrasound history? I remember you mentioned something when you spoke to us at the Fall for Tau event. So I thought maybe you can tell the story about how it was invented.
Ultrasound initially was invented by the need to detect things underwater. It was really encouraged by the sinking of the Titanic, because one of the reasons that the Titanic sank was because not one of the reasons, the reason it sank was because it clashed into an iceberg. And so when it clashed into an iceberg, the reason you couldn't see it is because icebergs tend to be about 90% underwater and only the tip is seen. And when it's dark, you can't see it. They weren't able to evade it and ended up crashing into it. And so immediately after that, a patent was filed on an idea to try and detect icebergs with ultrasound. And this was initially the rationale behind it. And soon after, there was a sonar trying to detect submarines instead of icebergs. And then from there, it started.
Okay, yes, that was exactly it. I wanted you just to share with the audience because I thought that was a really interesting fact. So I'm actually going to thank you both for your time. I think somebody in the Q&A also asked if there will be a recording. Yes, there will. It will be sent out to participants who registered. But let me just take a moment to thank you, Mary and Tali, for sharing your time and having this really interesting conversation, both about everything from, your research, which is in itself very, very fascinating, and also how you do it and how the lab works and all these other pieces that, maybe people might have not been aware of. So I certainly learned from it. I also want to take a moment with my board member hat on to remind everyone that Tel Aviv University is undergoing a campaign currently that's called the Uniformed University Campaign. There are still over 2,500 Tel Aviv University students, 35% of which who are women, serving in the reserves, and all these students, about 4,000 students that are transitioning back to their studies. So these are the future leaders of Israel. These are the future people who are going to make this country, lead the country and be the future researchers and hopefully, cure many other diseases. So we must support them. They're risking our lives to protect our beloved country, and it's our responsibility to help them fulfill their true calling to become the future leaders of Israel. So please join us in contributing to that campaign. In your follow-up materials, I'm sure that we will provide a link. But also you can go to the website, www.aftau.org, for more information. And then thank you to our friends from around the world for joining us. I know that people called in from everywhere. If you'd like to learn more about the work happening in Dr. Ilovitch's lab or across the university, that same website, aftau.org, will help you. A copy of today's conversation, like I said, will be in the will be sent to those who registered, but also in the webinar library found on the website. So with that, I want to wish you all a good rest of your day. Thank you so much, and
Thank you.
see you soon.
Thank you, Mary, also.
Thank you, Mary.
Thank you, Maya.
Thanks.